to talk with you about drug product quality today. And to start off, I have this comic to show, and some of you may be familiar with it. It shows a woman with her dog dropping off prescriptions at the pharmacy, and she says, "I'm dropping off prescriptions for both my dog and my husband. Give brand drug for my dog, but generic is okay for my husband." Yeah. And I feel like this is a perception that, you know, it's our responsibility to ensure that the public does not have this perception. We are responsible for producing and regulating the quality of drug products that the U.S. marketplace receives. So we have this common goal, the FDA and you as the industry, to produce good quality drug products. And I hope as we work together through events like this and then also communications through the application process that you don't have the perception that this is our relationship. So an overview of my talk, although my colleague already discussed the IQA process, I want to touch a little bit on where the drug product quality assessment fits into this IQA team-based approach for review in OPQ. As an assessor myself, I want to provide you with my own thoughts on key considerations for you as you're preparing your applications. And specifically, how do those key considerations apply to various parts of your application? I'll touch very briefly on some major deficiencies that we see and then concluding message. So as part of the overall quality assessment, there are multiple subdisciplines involved. Drug substance, drug product, and labeling are all handled by the Office of Lifecycle Drug Products, and that's the office in OPQ where I'm located. Manufacturing process facilities and microbiology, it's handled by the Office of Process and Facilities. Biopharmaceutics review is handled by the Office of New Drug Products. And you'll notice that there's an asterisk by drug substance and then also by the Office of New Drug Products. And that's because much of drug substance quality assessment is actually done by the Division of Lifecycle API in the Office of New Drug Products. And my colleague Ram will be discussing that next. So the focus of my talk today, there's supposed to be a color change here, but sorry, the top bar is supposed to turn from blue to orange. And I'm going to be discussing the drug product quality assessment, which occurs in OLDP. So a couple of things. I was a primary reviewer in OLDP and now a secondary reviewer, so I have experience with various dosage forms in the drug product quality assessment. And something to point out is the fact that OLDP is organized based on dosage form. So the team that reviews an injectable product is going to be different than the team that reviews a transdermal system or a modified release solid oral system. So when individuals are reviewing the drug product quality of your application, they are doing so with a frame of that particular type of dosage form, and therefore they're better equipped to help identify CQAs or critical quality attributes that are specific to that dosage form. My key considerations to share with you is, number one, meet all regulatory requirements in your submission. And I know that's a pretty big bullet. I feel as though many of the presentations that have been given already cover many of these regulatory requirements. And I also want to refer back to this same event that took place in 2017. All of the slides from that event are available online. And the talk by the same title given by my colleague, Guoping Sun, that actually outlines many very specific requirements that would be important to note. So please go back and refer to those old slides. But today I really want to talk more about some typical CQAs that we find lacking within an application. And this is my advice. Demonstrate product understanding in your application. Demonstrate that you have mitigated risks to product quality. And very importantly, provide supporting data. And I have a comic here that represents this. There are two people up at a chalkboard, and it looks like they're deriving an equation, and they seem to get from point A to point B just with no explanation. They say, then a miracle occurs. And although this is an exaggeration, we see this a lot. We're looking through a pharmaceutical development report. The applicant has provided risk assessment tables, and they go from having a risk assessment where they say particle size distribution of an excipient, it's a high risk, and then 2 pages later, without any explanation, it goes to a green low risk. We're going to have to follow up on that as reviewers at the agency, and we're probably going to have more questions because we see this discrepancy, and what are they potentially hiding? So really, providing us with supporting data to better understand how you've mitigated potential risks to your application is very important to help us ensure that we can help you get to approvability faster. So with that in mind, I want to touch on some CQAs that are in various parts of the application that we review during our drug product assessment. And the first is drug substance. So drug substance is reviewed by a DMF review team typically because the drug substance information is in the DMF, not all in the ANDA submission. However, as part of the drug product quality review, we are reviewing the drug substance and other raw materials for the material attributes that actually contribute to the final product quality. So examples of this would be polymorphism. As shown for zirconia here, you can see it has different polymorphic forms. Particle size, solubility, chemical stability. It's important that you understand how these various material attributes actually play into the finished product quality of your product. And also, when you're qualifying your suppliers, make sure that you communicate your requirements for quality to those suppliers and that you have an understanding of what they can provide to you. Risk to product quality. Solution for injection. These CQAs that I have shown up here, not all of them will apply to a solution for injection in the same way as it would a modified release tablet, where maybe polymorphism and particle size is very important for the tablet, but it might not be as important for a solution injectable where the drug substance is completely dissolved in that vial. But again, as a drug product quality assessor, you need to provide us with the information necessary to demonstrate whether or not any of these material attributes that are shown here are critical, and then how you propose to control them to end up with the final product quality. Another aspect is the design and formulation. As noted in previous presentations, differences in the design and formulation between the RLD and your proposed product may be allowed, but those differences could increase the risk. So it's important that you demonstrate that those differences don't affect the quality or safety of your product. Include considerations of size, shape, and visual differences, as well as considering the patient. Have the patient in mind. And a very typical example of this is tablet size. If the RLD has a 200 milligram size tablet and you're proposing a 1 gram tablet, obviously we're going to require you to provide us with a justification for that. Control of excipients. This is very important. USP monograph testing is a minimum requirement. Sometimes the tests that are included in a monograph are insufficient to control the material attributes of that excipient for your finished product. So typical material attributes could include particle size or viscosity. It's important that you communicate, just like with the drug substance, you communicate with the supplier to ensure an understanding of how they're producing that material and the lot-to-lot of variability that might be expected in that material, so that you can ensure proper quality of your finished product. So you may need to include tests that aren't included on the COA of your excipient supplier. For instance, particle size distribution, polymer viscosity, and then other tests including certification for BSE, TSE, melamine contamination, or tests for other impurities. For the control of drug product, I think it's been said many, many times, you cannot test quality into a product. So really, finished product testing alone should not be relied upon to ensure quality, which is why I emphasize some of the points about material attributes of your incoming materials. Also, although it's not a focus of the drug product quality review, we do interface very closely with the manufacturing process reviewer to determine how the raw materials and the formulation come together and are manufactured to produce a product quality that should be adequate and safe. So I'm not going to touch too much on overall specifications. Again, I refer to previous presentations, but I want to highlight impurities. Impurities is something that we spend a lot of time reviewing and we have to issue many deficiencies on. We have a new map that just came out. I encourage you to look at this to help you in establishing your impurity specifications for related substance impurities. And then also look to the ICH guidance, which includes how to control for residual solvent impurities, as well as the new elemental impurities, which I note even Cook highlighted in her keynote presentation as being really critical to ensuring that we help you achieve first cycle approvability. And I'll also caution that if you need to submit farm tox data in order to support an impurity limit, that will probably significantly alter our assessment timeline. So it's best if you know that farm tox data is needed, that you submit that in the original application to give us as much time as possible to review it. Analytical methods is something we also spend a lot of time reviewing. These 3 bullet points. Referencing compendia or DMF alone is insufficient in order to provide us with sufficient information to know that you know how to perform the testing and that test method is adequate for the intended use at your site. So make sure to provide the standard test procedure for all testing. We are not allowing you to simply say, we test per the monograph, but instead we expect a full standard test procedure with all of the testing procedure outlined so that we can understand what steps is the analyst actually taking to do that routine analysis. Also provide validation, verification, and transfer reports. And then lastly, although it's not common, we do see the importance of assaying not only the drug substance, but also critical excipients. So an example of this would be a penetration enhancer for a transdermal system. That assay, although it's just for an excipient, not for the drug substance, it does play a very important role in ensuring proper delivery of the drug. So in that case, we may ask for you to include extra testing in your finished product specification for that excipient. Container closure system. Is it appropriate to ensure quality over the proposed shelf life? Safety, function, performance, compatibility, and I've outlined some specifics here. We're looking for chemical stability. If your drug substance is known to oxidize, have you included an oxygen scavenger? Physical stability. Does your product need any kind of physical protection? In which case, maybe you need to include it in a blister pack instead of a bottle. Compatibility and protection from contamination is a big one as well. Glass delamination needs to be provided for injectable products, depending on the risk assessment that you provided. And then also, importantly, extractable and leachable assessments for many products is necessary. And the extractable and leachable assessments, that is something that's very important to provide in your original submission because and any supporting farm tox data. Because what we're finding is not only is the extractable and leachable not provided, we ask for the extractable and leachable, you provide it back to us. We find that farm tox data is needed due to the impurities that you see, so we have to send another deficiency, and it becomes a very drawn-out process. So the more information and justification and supporting data you can provide us in the original submission, the better off we'll all be. Also consider in-use stability issues that may require additional data. So this would be for very high-risk stability products where the act of just opening the bottle repeatedly throughout the intended use may have consequences for stability. And this is not a routine deficiency that we would issue, but again, a risk-based consideration. And I've highlighted here container closure system. Again, typically for oral products, what we often see is that the RLD is supplied in a single container, maybe a 30-count HDPE bottle, as shown on the right-hand side. But then the ANDA applicant is asking for a 500-count bottle, as shown on the left. As a quality reviewer, we're going to be looking at what is the void volume, what are the additional components that may be included inside of that packaging. Is the packaging appropriate given the intrinsic stability or instability of the drug substance? And a case example I have of this is where we requested samples for an ANDA, and the applicant provided us the large bottles of this gelatin capsule that contained beads. When we received the bottle, we found multiple fragmented capsules inside, and the powder spilled everywhere. What this particular manufacturer had done is that due to potential hydrolysis of the drug substance, they had included a large amount of desiccant. And they included desiccant in very heavy canisters in the bottle. They also had a lot of headspace in the bottle. And the desiccant was too much for the product. It had prevented the hydrolysis, but it dried out the gelatin capsules to the point that they became very brittle. Desiccant canisters, brittle capsules, a lot of wiggle room during the shipment process. Suddenly we started seeing these broken capsules, and so we had to ask the applicant to address it. So for finished product stability, poor stability we find is often the result of lack of product understanding. And we expect accelerated and long-term stability, 6 months at filing, but in addition to that, photo stability. And photo stability should be done per ICH Q1B, which will include not only appearance testing, assay of the drug substance and related substance impurities, but for specific dosage forms, it should include in vitro release testing as well. In-use stability, as I mentioned, may be needed on depending on the risk of the product. And then very importantly, we find the need to go into multiple cycles of review when the final specification for the stability results that have been provided aren't consistent with our additional recommendations that are needed. Examples of missing test results being physical stability, if polymorphism is an issue with your product, or in vitro release testing dissolution, where you didn't provide in your original submission a dissolution test that was adequately discriminatory, and so you had to revise that method. So now you and we have to wait for you to get additional stability time points and test with that final dissolution method. We also, as part of drug product quality, evaluate CMC information in the labeling. So chemical structure, ingredient list, and any USP monograph labeling requirements in the description section, as well as storage conditions and container closure system packaging information that may be provided in the how supplied section. So again, just looking for consistency across your application. Major deficiencies include missing toxicological studies, unacceptable physical properties, need for full-term stability data, failure to identify or include CQAs such as polymorphism, insufficient data to support use-related risk analysis, insufficient data to support drug-device compatibility, and then lack of safety assessment again, but specifically for extractables and leachables. So this is not a comprehensive list, but I do have a list of guidances for you to refer to. And the bottom one you see is a list. It's the it has major deficiencies in it. It's the FDA guidance for industry and submissions, amendments to ANDAs under GDUFA. And there you'll find major deficiencies not only for drug product quality, but for all of the disciplines associated with ANDAs. And so with that, I just want to close by saying again that we have this common goal. We all want quality drug products on the U.S. marketplace, but we need you to submit us the data to ensure that your product can deliver the therapeutic benefit that's indicated in the label. Please show your work. And so with that, I'll thank my division director as well as office director for giving me the opportunity to talk today.